This is the presentation for our paper, Towards Semantic Scene Understanding for Fine-Grained 3D Modeling of Plants. This is a work by Mohamad Qadri, Harry Freeman, Eric Schneider, and George Kantor. All authors are with the Robotics Institute at Carnegie Mellon University. There is a need for advances in agricultural robotics. Robots can help mitigate the effects of global population growth and expected food and labor shortages by automating tasks such as pruning, harvesting, phenotyping, and plant modeling. However, this automation is currently hampered by the difficulty in creating high-resolution 3D semantic maps in the field that would allow for safe and reliable manipulation and navigation. One reason is due to the low performance of traditional feature matching algorithms in agricultural settings. In this paper, we leverage environment-specific constraints to alleviate some of the 3D reconstruction and data association challenges. Specifically, we observe that robotic navigation in agricultural environments typically exhibits a specific pattern as seen here. Robots traverse fields one row at a time, generally moving in straight lines, and capturing images from side-facing cameras. This pattern of movement leads to specific constraints on the semantic features, in other words, the sorghum seeds, between the images. Specifically, we observe that the sum of 2D Euclidean distances between each seed and its neighboring seed should remain relatively constant. For example, the sum of distances from the circled node in right to its left neighbors is approximately the same in both images. Same is true for bottom neighbors, right neighbors, and top neighbors. We construct the following function, which captures the cost of associating a seed in image A to a seed in image B. The cost measures how different the neighborhood of the seed A is to the neighborhood of seed B in terms of 2D distance for each of the four regions. From this, we obtain a linear sum assignment problem, which we solve using the Hungarian algorithm. Now that we have a way to associate sorghum seeds between images, we use a standard SLAM formulation to obtain a final 3D reconstruction of the sorghum range. For the experimental results, we focused on testing different algorithms on real image sequences of 8 sorghum ranges. Sorghum fields are composed of rows, and each row is composed of several ranges, displayed here in yellow. A range is around 4 meters long, and each range may contain a different variety of sorghum. Empty spaces with no plants, about 1.5 meters long, separate consecutive ranges. Here are the mapping results. For our quantitative metric, we use the maximum distance mapped metric, which we define as the maximum distance that a SLAM system was able to map before a failure occurred. Failures can be due to reasons such as erroneous matches or a small number of detected features. In this table, we show that the maximum distance mapped in meters that was achieved with each method. Each row corresponds to a range. The first 4 columns show the results with traditional features, and the last column shows the results with our system. Using our proposed feature matching algorithm, we can map 3 out of the 8 ranges completely, and map around 65% of the remaining 5 ranges on average. SIFT performed the best out of the remaining 4 algorithms, with which we were able to map around 38% of the ranges on average. Finally, we compare the performance of our SLAM algorithm against OrbSLAM 2, a state-of-the-art feature-based SLAM system. We noticed that OrbSLAM 2 failed to map more than 0.35 meters, since a very few key points could be detected and matched robustly across frames. Here we can see some images illustrating this SLAM process and showing a final reconstruction. The first image on the top shows examples of semantic matches between 5 consecutive frames. The image below shows a 3D reconstruction along a range. The yellow dots are seed points in 3D. One of the key purposes in mapping and imaging sorghum is to get a yield estimate for the sorghum, counting seeds and/or predicting crop weight. However, images taken from a tractor driving down a row are fundamentally limited to capturing the side of the panicle facing the center. We are exploring counting improvements that can be made by imaging all sides of the sorghum panicle to get a more complete seed count. In order to use these images fully surrounding the panicle, we present a reconstruction pipeline. The pipeline segments seeds, then uses ICP to adjust relative frames and build a high-quality accumulated point cloud. We show how using sorghum seeds as semantic features can help create a more accurate 3D reconstruction. We first segment seeds, then register the 3D projected centers from multiple views using ICP. The original RGB image is on the right. We compare the result obtained with our method, center, against the 3D reconstruction obtained from performing ICP on the full point cloud, left. 2 noticeable reconstruction weaknesses can be seen. In the leftmost image, the stem is noticeably more fragmented, and the seed surface is more muted with lower brightnesses. This darker, more muted color indicates that frames have been mapped to each other with more noise, blending the original colors. We are working on quantitative measures for measuring the reconstruction quality. Thank you for listening to our talk.